In topic 33, you'll get an introduction to modern genetics. Okay, so we're starting in chapter 13 now, and the chapter starts off with a little bit of history of genetics. Inheritance has always been a mystery to people, and for a long time there were all kinds of different theories about how it worked. The prevailing theory in the mid-1800s was what they call blending inheritance, in which a liquid from each parent, plant or animal, blended to give blended characteristics. But there were lots of observations. For example, a couple would both have brown hair, they'd have a baby with red hair that maybe the grandparent had, and it just didn't fully make sense. Charles Darwin didn't accept this theory. He understood inheritance being very important for his theory of natural selection, but there really weren't any details to understand exactly how the patterns of inheritance worked. Well, eventually, a monk, Gregor Mendel, an Austrian monk, he was trained in mathematics and also in plant breeding. He started working with, of all things, a garden pea. And these plants are normally self-pollinating, but he was able to literally take pollen from one and fertilize it another and control the fertilization. And for years he observed the results and wrote all of this down, and he eventually came up with 2 laws of inheritance, which to this day hold true. Here's a picture of him, Gregor Mendel, the father of modern genetics. Now you have to realize that he was a very lucky guy in his choice of experimental plants. For one thing, pea plants produce lots of different offspring and reproduce very quickly. Had he chosen something like, say a tomato plant, or worse yet, a pine tree, it may have taken years for him to get any kind of results. But because he was working with these pea plants, he could have lots and lots of offspring, very fast generations, and so he could really get more data in a shorter period of time. Another very lucky thing is that the pea plants have a lot of observable characteristics that showed simple dominance. In other words, a lot of the genetics problems that we will be doing in this class and talking about are cases of simple dominance, and it's easy to see how one pattern will obscure the other, or one gene will obscure the other gene, and how this works. But there are lots and lots of things that we inherit that just aren't that simple, and we'll also refer to those as well. On the next page or two, I'll show you some of the pea plants' characteristics, and because of all of those, he could actually see these patterns emerge. He published his work in 1866 and died in 1884, and really during that time his work was not really accepted or understood by very many. So it was much later that he eventually became known as the father of modern genetics. Here's the picture of some of the traits that these pea plants have. And again, don't worry about numbers or ratios or anything like that. But seed shape, seed color, pea shape, pod shape, pod color, flower position, stem length, all of these things were cases of simple dominance where one would mask the other trait. And so because of all these different traits and all the breeding that he did, he came up with some very realistic, very accurate assumptions. We're also going to go through here, before we go over those, some terms used in modern genetics. And this is a little bit dry, but hang in there with me. And again, you may not completely understand them as I give them to you here, but as we work through the chapters, the upcoming chapters, it will all make sense, and you can go back and use this as a source. Okay, so first of all, genes. Genes are what we call heritable units of information about traits, and parents transmit genes to offspring. The genes are located at specific locations on specific chromosomes, And we'll talk a little bit later about how human chromosomes have been mapped, and many of these genes are now able to be identified on which chromosome they're found and which place on that chromosome. Locus is the word for the location of those genes. Number 2, cells with a diploid chromosome number have pairs of genes on pairs of homologous chromosomes. And we've already talked about this a little bit. A diploid cell has the 2 sets from each parent, and although the alleles may differ in what they're coding for, it's the same type of information. Once again, my terrible example, eye color. It would be located on this, if it were just one gene, located on the same place on the same chromosome, but one parent might be for blue eyes, the other for brown eyes. A mutation is a permanent change in a gene, and it could cause a trait to change. Sometimes these occur where, for example, a gene for flower color may be purple, and all of a sudden there's a mutated form that specifies white. These are the alternative forms of a gene. It is assumed that at one time they were all the same, but then a mutation caused the different expression and or mutated form, different gene, and those would be called the alleles. There's more. Yes, there's more. All members of a lineage that breed true for a trait have identical alleles for that trait. And what do we mean by breed true? It means if you keep breeding them, one offspring, another generation, if another never comes out, another gene expression, then it is likely that those have identical alleles. Now, if you cross those two with 2 different forms of a trait, for example, 2 flowers that have bred true for red and 2 flowers that have bred true for white, same species, different colors, and you breed those, what you'll get is what is called a hybrid. And so these might have gotten one trait from the red and one trait from the white. And again, we'll talk about this. This would be called heterozygous, which is here in number 5. So an individual with non-identical alleles, again, let's say one for red and one for white, would be called heterozygous. If they have identical alleles, like red and red, or white and white, that is homozygous. Hetero means different, homo means same. Now, if an allele is dominant, what we call dominant, that means its effects will mask the effect of the recessive pair. And we often use capital letters to signify dominant ones and lowercase letters to signify recessive ones. Again, as we move forward, a lot of this will soak in as we actually apply this. There's even more! A homozygous dominant individual has a pair of dominant alleles, such as shown by 2 capital letters. A homozygous recessive would be the 2 lowercase letters. Heterozygotes, which are sometimes called hybrids, a heterozygous individual would be shown by 1 capital and 1 lowercase. Gene expression is the process by this information on a gene or in a gene is converted to the actual physical or functional part of the cell or the body. And so these gene expression will determine the traits. To help keep the distinction clear between genes and the traits they specify, we use the word genotype and phenotype. Genotype are the actual alleles, such as, the big A, little a, or big A, or whatever. The phenotype is literally how they look. For example, red flowers. Okay. We also have F1 and F2, and in other words, some abbreviations here. F1 means first generation of the offspring. Parents is often abbreviated with a capital P. F2 means the second generation of offspring. And F is just an abbreviation for the word filial. Sometimes more directly it means sister, but it means, or females or daughters, doesn't mean sister at all. That was a complete lie. Filial means daughter, but it also simply means offspring. In this case, it refers to offspring. Okay, so this is just an example showing two homologous chromosomes, and the gene locus would be the area where the genes are found. They can be identical or not. In this case, the two genes, they're just showing them by colors here, would both be the same. They're both green. So that could be, I'm just making this stuff up, big G, oops, big G, big G. Okay? These look different. I don't know what they are coding for. It's a good guess, but they are a different color. So this might be big D, little d. And then these are just three more pairs of genes that are closer together down below. In summary here, I also want to explain again the words genotype and phenotype. These are examples of genotypes. The big P, this would be homozygous dominant, and we're saying that that P stands for purple. The recessive trait, we're going to use a little p, and this, again, the little p would be the genotype. The phenotype would be white flowers. How they look. Pheno means to show. Okay, then we have big P, little p. This is heterozygous for these the gene of color, flower color. And because P is dominant, the flower, the phenotype, will be purple. So again, as we move on through these topics, I think all of this will start to make sense, but this was just a little list we had to get through so that we could proceed into the other topics.